Thanks, everyone. My name is Will Salerno from the Regeneron Genetics Center. A couple of disclosures. Regeneron Genetics Center, fully owned subsidiary of RPI, and I do some other nerd stuff on the side. Today, am I hitting the wrong button? There we go. Who's saying that? Jeez. All right. Regeneron Genetics Center, part of Regeneron Pharmaceuticals. What's the bottom line here? I don't know if you caught Len, our co-founder at JP Morgan. He was talking with Jim Cramer after JP Morgan last week, talked about how you can't do large-scale drug discovery without genetics, without genes, without gene therapy. That's the future. Anyone familiar with drug discovery knows how expensive it can be, what the cost of failure is, and how we have to bear that. So the idea is what you see at the bottom here. You cannot pursue modern drug discovery without incorporating human genetics. What does that mean for the Regeneron Genetics Center? It means that slides are tricky and things pop around, but you get the gist, that we have to discover targets, that if we throw in a lot of genetic data, that helps us refine the targets that we want to find, that lead to precision medicine. And Regeneron Pharmaceuticals has built this great pipeline to do that, and the Regeneron Genetics Center feeds that pipeline by doing large-scale human genetics. So I'm about 5 years at Regeneron Genetics Center, and I'll say the biggest thing that was different for me coming in from academia wasn't the scale. I was used to scale. What I wasn't used to was the speed, because it's not just the competition you have in pharma that makes you want to go fast. It is the fact that we are trying to drive this all the way through to medicine. And obviously we want to make these discoveries, all this work that we do, translate into helping people and human health as fast as possible. So when we think about the speed, that led to a lot of the fundamental decisions of the Regeneron Genetics Center, which was we're going to do our sequencing with exomes, because we can do exomes fast, we can do them at scale, and we can translate that into discoveries quickly. However, a lot of times we get caught in this whole exomes versus genomes thing. I don't care about that anymore. There's a nice paper out from some colleagues at the Regeneron Genetics Center. It's on medRxiv now. It'll come out in one of the fancy journals at some point soon, talking about the differences between exomes and genomes. But what I want the takeaway to be is that there's sequenced genomes, there's sequenced exomes, and then there's imputation. And one of the things that we have found is that you can use imputation to do a lot of the whole genome variant discovery without having to sequence the whole genome. Okay, so one of the recent innovations led by John Overton in our lab, and this is some analysis done by people on my team, Tommy and Olga, that we switched recently from doing orthogonal, chip-based arrays to a capture-based imputation backbone. It's that middle thing there, the GXS, genotype by sequencing. It's available as a SNP diversity array product. And the idea is we can run one capture, our exome and our imputation backbone. We can sequence that one assay per sample, and then we can do a pretty good job imputing, and we compare this with a couple of the other arrays that are out there. So in order to do this very large-scale sequencing and get all the variants that we want for discovery, we do this single assay. However, we know that getting the imputation backbone is only one part of the whole scenario. You have to impute against something, and using the best publicly available resources can be very good in certain populations and not as good as we'd like in other populations. So the question is, how could we rapidly build out imputation panels that would allow us to, for every sample cohort that we get in, get the type of whole genome variants that we're really after? We need a readily accessible source of these globally diverse genomes to improve imputation across all of our cohorts. It's hundreds of cohorts. So we've been working with Ultima to evaluate the UG100. We were an early adopter, and we have seen, and I think you've seen this from everyone who's spoken so far, just how much the platform and the quality of the data has progressed. This is what we've observed with the progression, with the SNP data getting better and better. This isn't just the work that's done on the machine and the platform itself. This is working and, being an informatics person, working really closely with the informatics team at Ultima. They've mentioned working with the deep variant team and seeing them extend that code and software really just to drive all of this. So we've seen the SNPs get better. We've seen the indels continue to get better. I'm not going to go into a ton of detail because I think the Ultima team and Stacy and the team did a great job describing what we're all seeing on the platform. The one point I'll make is at the bottom left for you, that when we're thinking about finding these putative loss of functions that are critical for target discovery, these indels really do matter. So we still want to be driving the quality of these variants as much as possible to use those for discovery. However, we really do believe that the Ultima, the UG100 data is sufficient and good enough to create a resource for our imputation comparisons. So as we've been thinking about what that looks like, we think about what our history with the machine has been and when we are going to be ready to start cranking through a bunch of whole genomes. So this time last year, we were seeing about 10% uptime of the machine. By November, 100% uptime, and that's what we've had since. And this is a lot of hands-on effort with the Ultima team, a lot of hands-on effort by the team at the RGC. Sarah Wolf leads that effort with us, along with John Overton. So as of November, we've had this 100% uptime. What do we do with it? Well, we're going to sequence all the 1,000 genome samples and see how that looks in terms of an imputation resource. I'll admit we're not done yet, but what I did for this talk is take a snapshot, and this is what I wanted to get to today. What we've done, and this is work again led by Tommy and Olga on my team, we took 781 of the 1,000 genome samples that were sequenced on the UG100, and we built an imputation panel. We turned this around in about a week of, less than a week of informatics and about 5 weeks of sequencing. And we compared to the reference 30x genome set. We downsampled everything to 15x. And this is what we see. I don't think there's much detail. It's sort of, you look at the Illumina data, you look at the Ultima data, we can impute the individual samples very similarly. And I think if there's one big takeaway, it's that point in the light blue. What this allows us to do is to think about every cohort that comes across the RGC. Like I said, there are hundreds of them. And this isn't just the RGC, obviously. It's whatever cohort you're looking at. Take whatever cohort you have, and you can take some subset of those and rapidly sequence some whole genomes and add them to your imputation resource, okay? And then you will improve the quality of the imputation. And I think one of the things that's hard to take away from this plot, so I'll pop to this one, we're looking here at the genome in a bottle control samples. And you can see that there's a bit of a difference, right? The ones on the top with the blue circles are the HG005 through 7, and that population is better represented in these 781 samples than the two curves below it. The point here being that if you're able to feed in samples into your imputation panel that represent the cohort that you would like to study, you're going to drag that low allele frequency part of the curve up and improve your imputation. That's really what's going to allow us to do these things at scale. And this is the bottom line for us. I think Stacy made a point that we are looking for applications that are hungry for reads. For us, this is something where we want to binge reads, right? What we want to do is we want to have a cohort come in, 100,000 samples, 1 million samples, whatever it is, take 10%, binge sequence 10% of those, 20% of those with whole genomes, feed them into an imputation panel, and then broadcast what we learn about that across the rest of the samples that we sequence. This is what allows us to go fast, and this is what allows us to go cheap, and something like a really inexpensive but high throughput whole genome is what enables that. So when we think about the future for what we're doing with the UG100 and the Ultima team, there was the one slide we had that had sort of like the cost going down. Glad had that slide where it was going like that. I would like to say one thing that's going up is the sequencer. Up until now, it has been in the basement of our building. It is now getting pulled upstairs with the rest of the sequencers on this like fancy room with a glass wall, and everyone who walks in gets to see it. So in that sense, the Ultima UG100 is going up. And we're excited, I think, to be able to continue to drive some of these binge sequencing efforts where we want to just grab them and punch whole genomes through as fast as possible so we can broadcast that across the other data into our standard workflow. And I think that is the bottom line here, is that that's what a platform like the UG100 is enabling for us. So thanks for your time, and I'll be available for questions later.